I'm back with another episode of CRISPR Corner. In our last episode, I showed you how to use our design tool to find guide RNAs for gene knockout. But what if you're looking for guides for knockdown via CRISPRi or overexpression via CRISPRa? The design tool can help with that too. Navigate to www.milliporesigmabioinfo.com. Like last time, you may need to select your location. Head back to the CRISPR design tool by clicking on the icon. This time, click over to the pre-designed database KO Activate Inhibit tab. This portion of the tool only works for human or mouse. If you aren't working in either of those species, I'll give you some tips later in this video. Select your target genome from the drop-down menu. Then type the gene symbol, gene ID, or alias of your gene of interest into the box, and select the correct entry from the drop-down that appears. Next, select which type of guide RNA you're looking for. This tool can search several pre-designed databases for guide RNAs for knockout, activation, or inhibition. You can learn more about each of these libraries by clicking on the small I circle. This will give you information on the publication or source of the guide RNA designs. For guide RNAs for overexpression, you'll want to leave only the CRISPRa box checked. For guides for knockdown, you'll want to leave only the CRISPRi box checked. Once all of your information is loaded into the tool, press Submit. Your results page will load with your guide RNA target sequences. From here, you can download those designs and/or copy and paste them into a guide RNA ordering platform. But what happens if your search doesn't return any results? Or if you're working in a species that isn't human or mouse? Does that mean you can't use CRISPRi or CRISPRa to perturb your gene of interest? Of course not. If that happens, you'll need to go back to the Custom Design tab. Then you'll need to use the Input Sequences function to design guide RNAs appropriate for CRISPRi or CRISPRa applications. For CRISPRi, we generally recommend designing your guide RNAs from -50 to +200 base pairs relative to the transcription start site. For CRISPRa, we generally recommend designing your guide RNAs from -200 to -50 base pairs relative to the transcription start site. Copy and paste those sequence ranges into the design tool and follow other applicable design rules. If your gene of interest has multiple transcription start sites, you'll want to ensure your guide RNA will target all of them, or use multiple guides. That's it for this episode. On the next episode of CRISPR Corner, I'll show you how to order and customize the guide RNAs you've designed. Until then, if you'd like personalized guide RNA guidance for your next CRISPR project, reach out.